A-Level Biology 9700, Paper 4, Variant 3, May, June 2024, question number 7 and 8. And question number 7 is, experiments were carried out to determine the effect of light intensity on the rate of photosynthesis of a species of the unicellular protoctist chlorella. A cell suspension of chlorella was used. Carbon dioxide uptake was used as a measure of the rate of photosynthesis. See, suspension of chlorella was illuminated at a light intensity of 3 lux for 20 seconds. The carbon dioxide uptake by chlorella was measured at the end of the 20 seconds period of illumination. The experiment was repeated at 6, 9, and 12 lux, and in a dark room. The suspension was maintained at a temperature of 20 degrees C. So the table 7.1 shows the results of the experiments. And here you can see the light intensity and total carbon dioxide uptake after 20 seconds, the rate of photosynthesis. So the light intensity was 0, 3, X, 9, and 12. And total carbon dioxide uptake for after 20 seconds at 0 was 0, and at 3 it was 20, and at 6 it was 44, and at 9 72, and 12 80. So the rate of photosynthesis per micro molecular per second. So it was 0.0 at 0, and at 3 it was 1, and at 6 we have to find out this one. So it is blank space is given there. So at 9 it was 3.6, and at 12 it was 4. So here I would like to tell you that the question is, use table 7.1 to calculate the rate of photosynthesis at the light intensity of 6 lux. Complete table 7.1 by writing your calculated value in the space provided. So how do we calculate this value? So the rate of photosynthesis is equal to the, so it is always in this way. That is, we have to calculate by this formula. I'm going to write this formula as in this way. The rate of photosynthesis, rate of is equal to the total carbon dioxide uptake divided by time. So according to this formula, the total, it is 44, and the time is 20 seconds. So after dividing 44 by 20, the answer is 2.2. So we will write 2.2 over here. So this is the answer which we have calculated with this formula. Now we will move towards the next question. And next question is, plot a graph of the data in table 7.1 on the grid in figure 7.1 to show the effect of light intensity of the on the rate of photosynthesis. Draw a curve and extend your curve to show what would happen to the rate of photosynthesis if the experiment is carried out at 18 lux. So we have to take these values, and with the help of these values, we will draw this graph by taking all these values. Now, students, I will try my best to draw this because I have to draw on the laptop screen. So I will, on one side, you can see on x-axis the light intensity is given, and on y-axis the rate of photosynthesis is mentioned. So we have to we have 5 values. So we have to draw this. First of all, the first value, which is between light intensity and rate of photosynthesis, it is 0. So we will make a cross at 0, and all values are 0 in this case. So the next value, which is about, it is about 1.0, and it is on 3. 1, 2, 3, 4, 5. So it will be in the center of this. So it will be about in this way. And the third value, which is on light intensity 6, it is about 2.2. So it is at 6, it is 2, and 2.2, it is about over here. So, and the next value, which is on 9. So 9 is in between the 8 and 10. So the reading is 3.6. So here I'm going on 9. 9 is there, and there it is 3.5, and it is 3.6. Nearly round about this, because we are drawing on our screen. And the next is at 12. So at 12, it is 4.0. So at 12, it is 4. So it will be in this way. Now we have to join these lines with the help of scale. So all of you will use the scale. First value was on, so it will be in this way. It will be in this way. Don't forget to use the scale. So next we have to uh, extend. Now we have to extend between 12 to 18 according to the question. So in this way, we will extend from 12 to 18. So you can see the graph. I have tried my best to draw it accurately. I think it is about 95% correct. Only when we choose the points on screen, so a little bit it is difficult, but it is correct. You can follow this. And next question, we have extended this. Now we have to explain the shape of your curve from 12 lux to 18 lux. So we have to give some suggestions for 12 to excellence. But here you can see it is, the curve is level off. So from 12 to 18, and due to this level of, I would like to say that the light intensity is not limiting at this stage. So because it is going straight in this way, so we will write light intensity is not limiting at this stage. Why it is not? Because you can see it is maximum light intensity which a plant can have for the process of photosynthesis. So but we can say that the carbon dioxide can act as a limiting factor. Because at this stage, the um, uh, experiment has been provided for the process of photosynthesis, the maximum light intensity. So after this, you can say it means the rate of photosynthesis is not increasing by increasing the temperature. So it means that it is going, it is levels off, it is going straight. So it means now more temperature or the more light intensity is not required for this process. So at this moment, we can say that the light intensity is not a limiting factor, whereas the carbon dioxide can act as a limiting factor, because the light intensity is maximum, whereas we can say that the carbon dioxide can be act as limiting factor at this stage. And next question is, in photophosphorylation, photoactivation of chlorophyll results in the synthesis of ATP. So describe how photoactivation of chlorophyll results in the synthesis of ATP in photophosphorylation. So first of all, I want to exchange this question. Then I will write on the given space. So photophosphorylation is the process by which light energy is used to generate ATP. So the energy currency of light, the ATP. So this process occurs within the thylakoid membrane of chloroplast and involves a series of steps. And during these steps, during the process of photophosphorylation, you know that the photoactivation of chlorophyll takes place. So how it takes place, that is the chlorophyll molecules within the photosystems 1 and 2 absorb lights. So the during process of photosynthesis, the first step is that the absorption of sunlight. So at this moment, the chlorophyll molecules, there is the absorption of sunlight by the photosystem 1 and 2. So the energy which is absorbed will excite the electrons within the chlorophyll molecules to a higher energy level. So the excited electrons are ejected from the chlorophyll molecules and are passed along a series of electron carriers embedded in the thylakoid membrane, or you can say the electron transport chain. So these electrons transport chain, they will release energy. So the energy released from the electron transport chain will be used to pump protons from the stroma uh, outside into the thylakoid membrane. So this creates a proton gradient across the thylakoid membrane with a higher concentration of protons inside the lumen. So this proton gradient represents a form of potential energy. So then protons slow down their concentration gradient from the thylakoid lumen back to the stroma through a process protein complex called ATP synthesis. So in this way, I have explained this in detail, and we have to write these points on the given space. So I will make it short and I will write the major important points. So first of all, I would like to say that the chlorophyll molecules within photosystems, 1 and 2 absorb energy, light energy, which excites electrons within the molecules to a high energy level. The excited electrons are from the chlorophyll a series of electron carriers simply I would like to say electron transport chain ETC in the thylakoid membrane. So as electron move through move through this electron transport chain, they release energy. The energy released the energy released from the is used to pump hydrogen or proton ions from the stroma into the thylakoid lumen. So proton flows down their concentration gradient back through ATP which catalyzes photophosphorylation ATP into ATP. So it was all about question number 7. We are moving towards the question number 8. And again, question number 8 is about figure 8.1 is a diagram of the mitochondria. Inner membrane and outer membrane has been shown over there and outline the roles played by the mitochondrial membrane in respiration. And you know, two membranes are there, the outer membrane and the inner membrane. So the outer membrane is smooth, whereas the inner membrane has many folds, which are called as crystes. Now we have to write down the role played by the mitochondrial membranes in respiration. So according to their role, I would like to discuss something. So, you know that the mitochondria are often referred to as the powerhouse of the cell. So, due to their role in cellular respiration, their double membrane structure play a vital role in this process. So, first of all, I would like to discuss about the outer mitochondrial membrane because they have labeled them differently. So, we will describe both of these membranes. So, first of all, you can say It is high, the outer, we are talking about the outer mitochondrial membrane. It is highly permeable to small molecules, allowing for the free passage of ions and metabolites. And secondly, it contains protein import pores that allow the transport of protein synthesized in the cytoplasm in the mitochondrial matrix. So, and then the inner mitochondrial membranes, it has many folds into it, which increases the surface area. So these folds are called as cristae. So you can say the surface area for electron transport chain complex and ATP synthesis takes place over there. So it is highly impermeable. That was the outer membrane was permeable, whereas the inner membrane is impermeable to most molecules, ensuring a precise electrochemical gradient across the membrane. So it houses the complexes of the electron transport chain, which transfer electrons from electron donors to electron acceptors. So this process generates a proton gradient across the membrane. So I think the question is of only of 4 marks. So this description, I think this description is enough for it. So I would like to write the answer in the given space. First of all, we will write about the outer membrane, and then we will write about the inner membrane. So outer membrane is highly permeable to small molecules, allowing for the free passage of ions and metabolites. And then it contains it contains protein import pores that allow the transport of protein synthesized in the cytoplasm into the mitochondrial matrix. Next is we are going to write about the inner mitochondrial membrane. Inner mitochondrial has many folds called cristae, which increases its surface area. Which increases its surface area for electron transport chain complexes chain complexes and ATP synthesis. So it is highly impermeable. It is highly impermeable to most molecules, ensuring a precise electrochemical gradient across the membrane. In this way, it plays a vital role. It performs all these functions. Beside this, there are other many functions you can say. It also provides a platform for the electron transport chain and ATP synthesis, crucial for energy production. So it also create and maintain the proton gradient that drives ATP synthesis. So in this way, I would like to say it play a vital role in respiration. So next question is, the shape and number of mitochondria are constantly changing due to fission. Fission occurs when one mitochondrion split to form two mitochondria, suggests reason why mitochondria uh, fission carry out fission. So fission means the process of dividing into smaller mitochondria. So there are many reasons for which these mitochondria divide, or the fission takes place in them, or they carry out fission. So first very important thing is that the cell divides. You know, when cell division takes place, so every cell needs the mitochondria for the production of energy. So mitochondria need to be distributed evenly to the daughter cells. So fission ensure that each new cell receives a sufficient number of mitochondria to meet its energy needs. Secondly, you can say for quality control, you can say the quality control, or you can say the damaged or dysfunctional mitochondria can be isolated through fission. So this allows for their targeted removal through a process called mitophagy, or preventing the spread of damage and maintaining mitochondrial health. And beside this, fission allow mitochondria to adapt the changes cellular energy demands. For example, cell requires more energy, it can increase the number of mitochondria than fission. So mitochondria constantly undergo cycles of fission and fusion. So fusion can create smaller, more dynamic mitochondria, while fusion contains for the exchange of genetic material and the repair of damaged components. So in this way, you can see mitochondria fission is a crucial process that ensures proper mitochondrial distribution. So here I would like to write the uh, importance of this fission. First of all, the first I would like to say is the cell division. Cell division is firstly cells undergo cell division So, and daughter cells need even distribution of mitochondria to get energy. So they undergo fission process. Fission. And secondly, these cells, some of the cells, you can say the cells which requires more energy than it can increase the number of mitochondria through fission. Well, the next is uh, guanosine triphosphate is a molecule that is used as a source of energy in some reactions instead of ATP. Guanosine is composed of a purine similar to adenine and ribose. Suggest why GTP can be a suitable source of energy in some reactions, and we have to discuss why it is so. Guanosine triphosphate is a versatile molecule that plays a crucial role in various cellular processes, including energy transfer. So, GTP can be a suitable source of energy in some reactions. So, sometimes, similar to ATP, GTP possesses high energy phosphate bonds. When these bonds are broken through hydrolysis, a significant amount of energy is released. This energy can be harnessed to drive endogenic energy requiring reactions. So interconversion with ATP, so the enzyme nucleoside diphosphate kinase can readily convert GTP to ATP and vice versa. So this flexibility GTP to serve as a alternative energy source when needed depending on the specific cellular requirements. Sometimes, the GTP is essential for the elongation and termination phase of protein translation. So, GTP is involved in specific steps of metabolic pathways like the citric acid cycle. So, the GTP hydrolysis can be coupled with other reactions to drive them forward. This energy coupling mechanism allow for efficient energy utilization within the cell. So the choice between ATP and GTP often depends on the particular reaction and its energy requirement. So in this way, we have to write. I have explained many points, but I would like to say that GTP, according to the marking scheme, I would like to say that the GTP is essential for the elongation. ATP, similar to ATP, ATP, GTP possesses high energy phosphate bond. High energy phosphate bonds, which when broken through hydrolysis, a significant amount of energy is released. Moving to next question, and C part is old or damaged mitochondria reduce the ability of the cell to carry out aerobic respiration and produce the ATP needed for the metabolic processes of the cell. So, suggest what happens to these mitochondria to allow the cell to maintain the same overall rate of respiration and ATP production. So, I would like to say that what occurs to these mitochondria, or simply we can say how cell deal with damaged mitochondria. So, so to deal with the damaged mitochondria, they are sometimes you can say they are broken down, or there are some functions as for example, the damaged or dysfunctional mitochondria can significantly, impair a cell ability to produce ATP. So the energy currency of the cell, to maintain cellular function, cells have evolved mechanism to deal with these compromised organelles. So first of all, you can see there is a cellular process where damaged mitochondria are targeted for degradation by autophagy, and some kind there are, you can say they are fused with the lysosomes. Lysosomal enzymes, they break down the damaged mitochondria, recycling their components, and then mitochondria constantly undergo fusion and fission events. So fusion allow for the exchange of healthy mitochondrial components such as DNA, protein between mitochondria. Fission can isolate damaged parts of mitochondria, and then they can be targeted. So if mitochondria function is really impaired, cells can switch to glycolysis, less efficient but faster way to produce ATP. So you can say if mitochondrial damage is extensive, it cannot be repaired, the cell may undergo programmed cell death. So this is a controlled process that prevents the spread of damaged mitochondria and potential harm to the organism. Cells can maintain, so in this way, the cells can maintain their energy supply and function despite and presence of damaged mitochondria. So these, you can say, uh, after I have given explanation, now I am going to write the damaged or dysfunctional functional mitochondria are engulfed or break down. I think it is titled to uh, only the breakdown broken down by lysosomal enzymes. And then mitochondria undergo fission undergo fission reaction to produce new bacteria. And I would like to write. So I think uh, fission can isolate damaged part of mitochondria, which can then be targeted. So in this way, the mitochondria dealing takes place. So it was all about question number 7 and 8. Please like, share, and subscribe my channel. Thanks for watching.